Thank you very much. It's a pleasure to be here and present to you all. And thank you for Oxford Nanopore for inviting me. So I'm going to talk to you a little bit today, switching gears from a lot of the talks you've heard of whole genome sequencing or whole exome sequencing. I'm going to talk to you a little bit today on targeted PCR amplification, enrichment, and using that for nanopore sequencing in the carrier screening market. So to start off, there we go. To start off, basically everyone is a genetic carrier for some condition or disease. There are over 200 different genetic disorders that have been described with autosomal inheritance. Recessive inheritance. And the prevalence is often population specific. Recently, in the past couple years, 113 genes have been identified for a carrier screening panel. There are many panels that are much larger than this, 400, 500 genes available from centralized markets or centralized companies. Basically, the idea of carrier screening is to identify variants in at-risk couples, so mothers who are expecting or planning a family, and if a variant is identified in the mother, then the father is tested. And if both carry a variant in the same gene, then there's a 25% risk that the baby will be born with that condition. And interestingly, there we go. Interestingly, 6 of the top 10 genes in carrier screening by prevalence are very challenging for short-read sequencing. And so typically a sample will go to one of these carrier screening labs and they will run a NGS panel on a vast majority of the genes in the panel that you pay for. And then a number of these particular genes are one-off panels using specialized methods that the carrier screening lab has to be proficient in carrying out. And so I've listed here a number of genes, including Fragile X and alpha thalassemia, hemophilia A, Ka, and SMA, that have different gene variation associated with these specialized panels that are required. And so what we've done is develop a single workflow that can address a lot of different genetic variation in some of the most prevalent diseases for carrier screening. There are, guideline updates that we are looking at. There are novel gene therapies for some of these genes that we're allowing our customers to get better answers on. There's an increased awareness for health equity, not just mostly white population, but a pan-ethnic population across the US and across the world that we're looking at. And we're hoping that this kit that we're developing will help decentralize the assay testing. So we're using PCR amplification with nanopore sequencing, and we've built our own software to do a lot of this testing across 11 different genes, CFTR, SMN, FMR1, HBA1 and 2, HBB. I'll be talking about all of those today in a little bit of a deep dive. I'm going to not talk much about any of the genes associated with mixes with mix D listed there. We use 4 different mixed tubes and barcode tagging, followed by ratiometric mixing of these different PCR amplifications. We're currently using LSK14 and R10.4.1. We are currently using Guppy, but we will hopefully be switching over to Dorado soon. And we will both support the Mark 1B and the Grid Ion. We're building out our own software package that incorporates, after the sequencing is done, we do live-based calling on the instrument or computer, and then importing that base called data into our software along with the sample sheet that delineates the sample and the barcode, and then analyzes for each of these different sequence variants. And so across the board, we've tested so far 589 samples with over 98% accuracy. These are cell lines and whole blood samples. I have listed here some of the variation that we're detecting and what we're detecting for each gene. So I'll go into a little bit starting with a fairly easy gene. This is easily detected by NGS. We incorporated some genes that were easy by NGS for some of the smaller labs, not these large centralized labs, but some of the smaller labs, if they want to start doing carrier screening in-house. We incorporated some of these genes. And I should note that these genes represent about 70% of the at-risk carrier frequency compared to gene panels that are much larger. So these smaller labs can start adopting this panel. So CFTR, pretty easy, SNV and indel prediction along with exon del dupe identification. SNVs are really easy with the 10.4.1 kit. Here we have two different haplotypes associated with G622D and T501I. The T501I has not yet been verified from Sanger sequencing. These are both blood samples that we have a collection of in-house. G622D is a variant that's more prevalent in Southeast Asian populations. It's not as prevalent in the white populations. However, and G622D was also not updated in the recent CFTR guidelines, but we do find it in pan-ethnic populations somewhat frequently. So pretty easy to do SNVs. We've also looked at exon del dupes, CFTR DELI23 and DELI19 and 20. These are more prevalent del dupes in CFTR. We're easily able to identify those. The orange line there on the what would that be? Your right side, or your left side, is DELI23, and it falls below the line listed here. That purple dotted line, that sample falls below that purple dotted line. All the other wild type samples fall above that, including a sample that has a CFTR DELI19 to 20 large exon deletion. It falls with the normals when you're looking at exons 2 and exons 3. When you're looking at exons 19 and 20 and exon 21, the DELI19 and 20, you can see that falls below the line and everything else is above. So across the board, we're got 100% variant agreement with our known expected variants that we've identified. And we looked at that 100% agreement is across 65 different unique variants, and it's pretty good performance. So moving up a little bit more complicated, we also are able to elucidate copy number evaluation. So, and SNV indels phased to the copy that's present. So in SMN 1 and 2, SMN 2 is different from SMN 1 by just one single nucleotide change in exon 7. There's also another variant, but it doesn't change the amino acid at all. This exon 7 induces exon skipping in SMN2, but not in SMN1, so you get the full-length gene. However, the number of copies of SMN1 or SMN2 will help tell you how severe the disease is. And so 0 copies of SMN1 can be overcome by multiple copies of SMN2. So you have to be able to differentiate these, and they're 99.9% similar. So we're able to align sequences to either SMN1 or SMN2 based on these long reads that we have across here, and differentiate them based on the C to T change in exon 7, and then count the number of copies of SMN1 and SMN2. And we are also able to associate SNVs. As mentioned, they're only about 5% prevalence, the SNVs are, but there are silent carrier variants and disease modifier variants that are not pathogenic, but help us to determine specific genotypes within here. So again, we're for copy number of SMN1, 0 or 1 copy, 100% effective in cell lines in whole blood. For other copy numbers, 2, 3, 4, it drops a little bit, but still above 70 or 97% across the board. So getting a little bit more complicated, we can do repeat sizing. FMR1 is a repeat assay in the 5' UTR. When repeats get above 55, you're a carrier, and when the repeats of these CGGs get above 200, it's a full mutation. We're not only able to look at the CGG repeats, but we're also able to look at AGG interrupts. And so these are very low complexity DNAs that we're able to amplify and then resolve both size and interruption status. And so here's a sample that's a 3082, so it would be a carrier mutation, and we align these sequences and then stack rank them so that we get a number of counts for the 30 CGG and a number of counts for the 82 CGG. You can convert that image, just turn it, and you get a histogram of the sizes of 30 and 82, and then we can phase on top of that the AGGs. And so we can see that there are 2 AGGs on the smaller 30 CG fragment, and there are 0 AGGs on the larger 82 fragment, and that's that increases a high risk for expansion. So across the board, we've been able to do this well past the 200 mark. Our cutoff for actual sizing is about 250, and then much larger, we can get up to 1,300 CGGs, which is about a 4,000 nucleotide fragment. We can see that and flag it. So we're able to get 100% sensitivity and specificity off of these. I'm going to get a little bit more complicated and layer on not only SNVs and indels, but structural variation and exon del dupe together in thalassemia with HbA1, HbA2, and HbB. These are all in the same tube. Most of the variation is due to one of the 4 copies of HbA1 or HbA2 being missing. And then there's, in the structure of hemoglobin for thalassemia, there's 2 copies of the HbA and then 2 copies of HbB that sit on top of that. And so we're looking at all of this in one tube. And there's a lot of structural variation in this alpha thalassemia region, and so we are able to not only identify the structural variation, but differentiate different variants, different structural variants with the TI and FIL and 3.7 DELs. And so we have a pattern matching for HbA, where we look at different regions within HbA. The HbA region, we do pattern matching across each of those different regions. This is a wild type sample. A sample with a dropout in the HbA2 region would look like that, and then a sample with a dropout across both HbA1 and HbA2 in this region looks like that at the bottom. And we do pattern matching of each of those to identify those. We can also look at SNVs associated with individual copies. Here's an SNV in HBB, and we can do copy number for HBB that's a little bit differently. We're only looking at 2 amplicons, exons 1 through 3 in that one. And so within this, we've gotten very good sensitivity and specificity. We have almost 100% sample agreement. There were 3 anti-3.7s, which is a triplication of 3.7 that's not pathogenic, but that one, we missed it in 3 different samples, and so it's still 100% ability to call carriers. We tested a sample set of 232 samples, and these are 232 presumed normal samples. We were able to identify 37 carriers. Most of these individuals had no idea they were carrying a variant. And so we're able to identify variants across the board in the streamlined panel. And this carrier frequency is similar to the expected carrier frequency for a panel this size. And so with this unified workflow, we're able to provide actionable genotyping and provide informed decisions for the lab that then feeds into the clinician that then benefits the patient. So thank you very much for listening. I've gone over a little bit. This is the research team. For Halloween, we did Bob Ross and the Multiverse. And then the larger team across Asuragen for our Halloween party. We have a huge team at Asuragen working on this particular project. A few of them are here today. So thank you very much for listening.